The AP Bio exam is coming up, and it's going to be full of tricky questions that are based on difficult concepts. To help you get ready, I've identified the most common pitfalls, traps that AP Bio students experience. In this video, I'm going to teach you how to identify these traps and avoid them so you can crush it on the AP Bio exam or on your teacher's next test. Because these pitfalls come from all over the curriculum, watching this video is going to be a great way that you can review for the exam. Before we get started, I wanted to let you know that I've put together a checklist that you can study for the AP Bio exam or for any content that you experience throughout the course. You can download the checklist at apbiosuccess.com/checklist. Pitfall number 1, don't underestimate hydrogen bonds. Hydrogen bonds stem from molecular polarity, and the molecule that's the king of molecular polarity is water. In this diagram of a water molecule, you can see that the electron sharing is unequal. The electrons are clustered on the oxygen side of a water molecule. That creates a slight negative charge over here and a slight positive charge over here. Slight negative charge on the oxygen side of the molecule and a slight positive charge on the hydrogen end of the molecule. Because of polarity, water molecules will form hydrogen bonds with one another. These are intermolecular bonds, intermolecular between molecules, and they form between the partially positive hydrogen side of one molecule and the partially negative oxygen side of another water molecule. Individually, the hydrogen bonds that connect water molecules are very weak. These bonds, these hydrogen bonds, are much weaker than the covalent bonds that hold a water molecule together or ionic bonds. But collectively, hydrogen bonds are extraordinarily important, and as we'll see, they structure so much of what happens in biology. Hydrogen bonds between water molecules creates cohesion. Cohesion means sticking together, and here you see a bunch of water molecules that are sticking together because of hydrogen bonds. Cohesion, in turn, creates surface tension, and that's the force exerted by the water molecules on the surface of a body of water, and it creates a kind of intermolecular web or net upon the surface. And of course, in this representation, the water molecules are shown vastly out of scale, but it gives you an idea of why you can, with finesse, float a paper clip on top of the surface of water in a cup. Here are two adaptations that are directly connected to surface tension. Water striders are insects that can stand on a body of water, and geckos are capable of running across a lake. Why? Because of hydrogen bonds and surface tension. Cohesion from hydrogen bonds also explains water's high heat of vaporization and water's high specific heat. Heat of vaporization is the amount of energy it takes to make water evaporate. Evaporation requires breaking those hydrogen bonds. High specific heat means the amount of heat required to raise the temperature of water by 1 degree. Because of hydrogen bonds, it's hard to get water molecules to move around faster and to increase their kinetic energy. Hydrogen bonds and cohesion also explains why ice floats. Ice floats because it's lighter than liquid water, and that's because of how hydrogen bonds create this spread out crystalline matrix when water freezes. That crystalline matrix is less dense than liquid water. Hydrogen bonds also result in adhesion. Adhesion is hydrogen bonds between water molecules and other polar substances. For example, the cellulose walls that make up the water conducting tubes of plants. And here you see that bond forming. And what's that all about? Well, in cellulose, there are all of these hydroxyl functional groups, and you can see that they're kind of like a water molecule. They're polar, and so water molecules can interact with these hydroxyl groups and stick to them. Adhesion and cohesion also explain things like capillary action, which is shown here, water molecules crawling up the side of a small tube that's made of some polar substance. And it also explains the incredible phenomenon of transpiration, which is how water moves up to the top of plants. What happens is that water evaporates from the pores in the underside of the leaves. Those are called stomata. As that does, that creates surface tension. Because the water molecules all cohere to one another through cohesion. As one water molecule leaves, it pulls the water molecules below. Those water molecules are also interacting by adhesion to the tubes that make up the water conduction system of plants, and that goes all the way down to the roots. Again, water, surface tension, adhesion, and cohesion, explaining so much of biology. But hydrogen bonds aren't just limited to water. You also see them, for example, in DNA, where they hold together the paired nitrogenous bases, adenine and thymine, cytosine and guanine. Here you see a hydrogen bond that's formed between this hydrogen atom and this oxygen atom, this hydrogen atom and this nitrogen atom, and here you can see those nitrogenous bases in the middle of the double helix, holding the two strands of the double helix together. You also see hydrogen bonds in the secondary, tertiary, and quaternary levels of protein structure. Secondary structure is all about hydrogen bonds, and in tertiary structure and quaternary structure, hydrogen bonds combined with covalent bonds and ionic bonds and hydrophobic interactions to form the complex shape of proteins. If you remember back to Unit 6, protein synthesis, hydrogen bonds are everywhere. Hydrogen bonds are how anticodons connect with codons. Hydrogen bonds are how tRNAs fold up into their very specific shape. Hydrogen bonds are everywhere. Hydrogen bonds also show up in Unit 3 when you talk about enzyme action. When enzymes and substrates interact to form an enzyme-substrate complex, the temporary bond between enzyme and substrate is often a hydrogen bond. In Unit 4, when you were learning about homeostasis, you should have noted that breaking hydrogen bonds is the basis of evaporative cooling. Sweat glands, which are shown at number 1, release sweat onto the skin, and the most energetic water molecules vaporize. That involves breaking hydrogen bonds. As those water molecules leave the body, they take away their heat, and that lowers overall temperature. That's evaporative cooling. Dogs are doing the same thing when they pant. The takeaway is don't underestimate hydrogen bonds. On an FRQ or on a multiple choice question, when you're asked what kind of bond does it work, it's often going to be a hydrogen bond because they're so omnipresent within biology. Hydrogen bonds are weak intermolecular bonds, collectively significant. They explain the key properties of water. They can also occur between oppositely charged parts of any polar molecule, and they're a key force in many biological phenomena: DNA structure, protein synthesis, transpiration, evaporative cooling, perspiration, etc. Your success in AP Biology starts here. Are you struggling with AP Bio? With Learn-Biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to Learn-Biology.com to find out how you can master your biology course and crush the AP Bio exam. Pitfall number 2. Plants don't just photosynthesize, they also respire. Plants are photoautotrophs, and they perform photosynthesis. Photoautotroph means self-feeder using light. Plants feed themselves. They create their own mass by pulling in carbon dioxide and water. Oxygen is the waste product. Photosynthesis happens in organelles called chloroplasts. And here's the equation, which you should know. Carbon dioxide and water are combined with light energy driving the process to create carbohydrate, C6H12O6, and oxygen. Photosynthesis is the source of almost all biomass in the biosphere. Here you see a pyramid of biomass. Producers at the bottom are doing their production through photosynthesis. It's also at the basis of almost every food chain. Here you see a terrestrial food chain, and here you see one for an aquatic ecosystem. But just like you, plants also respire. They take glucose, and in the presence of oxygen, they create ATP, and the waste products are carbon dioxide and water. And here you can see that same equation in the context of the entire plant. The carbohydrate was made through photosynthesis, but then when the plant needs that energy, it'll take that carbohydrate and in the presence of oxygen, it'll use it to create ATP and release carbon dioxide and water as waste products. In their mitochondria, plant cells produce ATP just like you do. That's life's universal energy carrier. Here's ATP, and the thing to remember is that all plant cells respire all the time, even while they're photosynthesizing. Non-photosynthetic plant cells don't have active chloroplasts, and they do have mitochondria. When you have a potato stored away in your cupboard, it's consuming oxygen, it's releasing carbon dioxide and water as it does cellular respiration. Deciduous trees which have lost their leaves during wintertime only respire. At night, plants only respire. And for AP Bio, you have to know that plants respire. For example, the Respiration with Respirometers lab is all about using plants, seeds in particular, as respiring organisms where you can measure their respiration rate. And you can do it in this old school way with respirometers, or you might do it in a probe that's measuring carbon dioxide production or oxygen consumption. But nevertheless, the point is that plants respire. Here's a graph and prompt about exactly this from the 2012 AP Bio exam, question 1. An agricultural biologist was evaluating 2 newly developed varieties of wheat as potential crops. In an experiment, seedlings were germinated on moist paper towels at 20 degrees C for 48 hours. Oxygen consumption of 2-day-old seedlings was measured at different temperatures. The data are shown in this graph below, which you can see right here. So your job is to calculate the rates of oxygen consumption in milliliters per minute for each variety of wheat at 7 degrees C and at 17 degrees C. Show your work, including your setup and calculation. We're not going to go through the whole thing because the point for now is that the seedlings are consuming oxygen because they're doing cellular respiration, even though they're plants. So again, you have to hold on to this idea. Plants do cellular respiration. And to talk a little bit about the rate, variety A's rate at 7 degrees. Let's find it, variety A at 7 degrees. It's these boxes over here. It's going to be 0.5 milliliters consumed over 80 minutes. And you do some math and you get 0.00625 milliliters per minute. And then if you look at variety B's rate at 7 degrees, so here's variety B, 7 degrees, find the circle, 7 degrees. We've got 3.5 milliliters consumed in 80 minutes. Do a little bit of math and you get 0.04375 milliliters per minute. Here's another set of data and questions about a similar topic. You could see this on your teacher's next test or on the AP Bio exam. Here's how this works. You take 20 seeds, you grow them in the light, you mask them first, and a week later you mask them again. You dry them out so that you're not measuring water uptake, you're just measuring the effects of either photosynthesis or respiration. And then you take another 20 seeds, you grow them in the dark, and again, you mask them a week later, and you're only measuring their dry mass. To keep things simple, I've set the initial mass equal, but it might be more complicated in an actual FRQ. Your job is to explain the change in mass in the group in the light and exchange the change in mass in the group in the dark. We'll start with the group in the dark because it's simpler and it relates to the theme of this pitfall. In the group in the dark, the mass is decreasing. It's gone from 35 to 18 because of cellular respiration, which releases carbon dioxide and water as exhaust products. In the group in the light, the mass is increasing because of photosynthesis, but the increase, this increase from 35 to 64, it shows the mass gained from photosynthesis minus the mass lost from respiration. So in other words, this is a net gain, photosynthesis minus respiration, and that would be important to say in an FRQ. Here's a graph from one of the many practice FRQs on learn-biology.com, but something very similar to this could easily be on the AP Bio exam or your teacher's next test. Your job is to explain what's happening at points 1, 2, and 3 in this graph. We're only looking at plant A for this question. A little bit of orientation. The x-axis is about light intensity going up to the right, and the y-axis is about carbon dioxide uptake, which is going up as you move up. What's happening at point 1? Point 1 is respiration only, and what you should note is that the CO2 uptake is negative. What does that mean? Again, remember, plants uptake carbon dioxide as a result of photosynthesis. They fix carbon dioxide and absorb it into their bodies. But what you're doing right now is cellular respiration, and you are outputting carbon dioxide, and that's what this plant is doing over here. Why? Because the light intensity is 0. There's no photosynthesis, so all this plant is doing is cellular respiration. At point 2, note that the carbon dioxide uptake is 0. How could that be? There's light, but only enough light to offset the output of carbon dioxide. So the amount of carbon dioxide that's being produced by respiration is being offset by the amount of carbon dioxide that's being absorbed by photosynthesis. And then at point 3, we have the maximum net carbon dioxide uptake. And remember, this isn't just about photosynthesis, it's photosynthesis minus respiration. What's the takeaway? You can avoid pitfall number 2 by remembering that plants do both photosynthesis and respiration. During photosynthesis, they're absorbing carbon dioxide and water as they produce carbohydrate. They release oxygen as a waste product. During respiration, plants are doing exactly what you do, but they're doing it with carbohydrate that they've made themselves. And what they are doing is respiring that carbohydrate in the presence of oxygen, producing carbon dioxide and water. Pitfall number 3, photosystem 2 precedes photosystem 1. To address this pitfall, let's start by reviewing where photosynthesis happens. It happens in plants, it happens in the leaves of plants, in organelles called chloroplasts. Within chloroplasts, photosynthesis occurs in 2 phases. The first are the light reactions. The light reactions convert light energy, which you see over here, into chemical energy in the form of ATP, life's universal energy carrier, and the electron carrier NADPH. In the Calvin cycle, the products of the light reactions, NADPH and ATP, are used to create carbohydrates. That's represented down here as the sugar. And super importantly, what the Calvin cycle does is it fixes carbon dioxide, converting it from a low energy gas into high energy sugars. The light reactions happen in chloroplasts. Here's a chloroplast here at M, along the membranes of thylakoid sacs. So here are the thylakoids, and here is just one of these really enlarged, and it's along the membrane of the thylakoid that the reactions of the light reactions occur. During the light reactions, chlorophyll, which is the key pigment involved in photosynthesis, is embedded in structures that are called photosystems. Here's a photosystem over here. It's mostly protein, and chlorophyll embedded in photosystems is able to convert light energy, here's light energy, into a flow of electrons, into electron energy. Here you see an electron energy that's being boosted from a magnesium atom within chlorophyll. During the light reactions, electron flow generated by 2 photosystems drives the creation of ATP and NADPH. This diagram shows a thylakoid membrane. Here's one photosystem, here's a second photosystem. This first photosystem is generating electron flow that's used to power proton pumping. That proton gradient later is used to generate ATP through chemiosmosis as protons diffuse through the ATP synthase channel and enzyme. And this photosystem over here uses its electron flow to reduce NADP plus into the electron carrier NADPH. The pitfall is associated with the fact that these photosystems were numbered in order of their discovery. So as electrons flow, photosystem 2 precedes photosystem 1. That is a fact that has enraged AP biology students and biology students in general for all of eternity, and now you're going to feel the same way, but you just have to memorize. Photosystem 2 precedes Photosystem 1. Photosystem 2's electron transport chain powers water splitting and ATP synthesis. Photosystem 1's electron transport chain, shown over here, powers the reduction of NADP plus into NADPH. So the takeaway, the light reactions are one of the most beautiful processes in biology, but the photosystems are named in order of their discovery. Photosystem 2 precedes Photosystem 1. It's a fact to memorize. Just get it done. Pitfall number 4, mitosis ain't meiosis. The two sound alike, but they're quite different, and you need to be on top of those differences. Let's start by quickly reviewing these two processes. Mitosis, its functions are growth and repair and asexual reproduction. It involves one round of cell division. The chromosome number stays the same. The daughter cells are clones of the parent cell. Notice that there are 4 chromosomes represented here in this parent cell, and there are 4 in each daughter cell, and they are genetic clones of the parent cell. For meiosis, the function is creating gametes. There are 2 rounds of cell division. Here's the first, and here's the second. The chromosome number is cut in half. Notice that there are 4 chromosomes over here in the parent cell, and there are only 2 each in each of these gametes. The daughter cells are genetically distinct from their parents and from each other. That is what meiosis is all about. Meiosis, to emphasize this point, is reduction division. Cells go from diploid to haploid. Step 1 is DNA replication. Here you see the parent cell, and here you see the parent cell with each of its chromosomes doubled as 2 sister chromatids. Creates doubled chromosomes made of 2 sister chromatids. Step 2 is meiosis 1, which separates the homologous pairs and creates haploid cells. So by the time we're over here, after the meiosis 1 division, the cells have gone from diploid with 2 sets of chromosomes to haploid with 1 set of chromosome. And in meiosis 2, the sister chromatids are separated, and the result is 4 unique haploid gametes. Meiosis generates genetic diversity in 2 ways. The first is independent assortment. The second is crossing over and genetic recombination. If these are unfamiliar to you, please watch my video, How Meiosis Creates Variation. Here's another part of this pitfall to avoid. When you're describing what happens in meiosis, you must specify whether it's happening in meiosis 1 or meiosis 2. So for example, anaphase 1 is when homologous pairs are getting pulled apart. Anaphase 2 is when sister chromatids are getting pulled apart. Prophase 1 is when crossing over is happening, and so on and so forth. You have to specify which division is occurring. Mitosis and meiosis both involve cell division, but their functions and outcomes are different. Mitosis is cloning cells for growth and repair. Meiosis is creating genetically unique gametes for sexual reproduction. They sound alike. As an AP Bio student, you need to know the difference. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com. You can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Pitfall number 5, positive in biology isn't good, negative isn't bad. The terms positive and negative show up in many contexts within biology, and we're going to talk about a few of them. The first one involves feedback loops. Positive feedback loops amplify a biological process, driving it to a conclusion. Sometimes these positive feedback loops are good. For example, the growth of a baby within the uterus over here leads to stretch receptor activation. That leads to a signal that gets sent to the brain that causes a gland to release a hormone that's called oxytocin. That oxytocin goes into the blood, it goes everywhere, it's picked up by receptors in the uterus, and that causes uterine contractions to intensify. That intensification leads to more stretch receptor activation. It's a positive feedback loop, and it results in contractions that cause childbirth. You are probably here because of this positive feedback loop. It is a good thing. But here's another positive feedback loop that isn't good. It's bad. This is called the extinction vortex, and it's a population genetics phenomenon. It's often caused by fragmentation of habitat, and that leads to small isolated populations. Because they're small and they're isolated, there's genetic drift and inbreeding that leads to a loss of genetic diversity, that leads to reduced fitness and adaptability. The population is then less able to reproduce. It has higher mortality, and the population becomes smaller. So it feeds back upon itself with more genetic drift and inbreeding. In this case, this positive feedback loop is actually a vicious cycle, and it contributes to the ongoing extinction crisis that we are experiencing right now. As opposed to positive feedback, which can be good or bad, negative feedback in biological systems is generally good. And these terms are a little bit confusing because of the way that we use these terms in common speech. When you get negative feedback about a project, that doesn't feel very good because somebody's probably told you that it isn't very good. Positive feedback, by contrast, is someone saying, that's fantastic. But in biology, things are different. Negative feedback is a condition in which the output of the system causes the system to return to a set point, kind of like your home heating system. You set the thermostat at the desired temperature. If the temperature falls below that, that activates the furnace. The furnace puts out heat that raises the temperature, and the system turns off at a more comfortable temperature. Negative feedback loops in biological systems are essential for homeostasis, which is maintaining consistent life-friendly internal conditions, and negative feedback loops are often organized into antagonistic pairs. As an example of how negative feedback systems that are organized in antagonistic pairs works, let's look at blood glucose regulation. You have a blood glucose set point when you eat a big meal, a carbohydrate-rich meal in particular, that will lead those carbohydrates and sugars to be absorbed into your blood. All of a sudden, your blood glucose level is above the set point. That will be picked up by certain cells within your pancreas that release a hormone that's called insulin. The insulin diffuses into your blood, and there are receptors in fat cells, liver cells, and muscle cells, not shown here, that lead that glucose to be absorbed from the blood, converted into glycogen in liver cells and muscle tissue, and converted into fat in fat cells. That drops glucose back down to the set point, and then the system turns itself off. When you've gone a couple of hours without eating, your blood glucose level starts to drop. That drop is actually picked up by other cells in the pancreas that release a different hormone that's called glucagon. Glucagon diffuses into the blood, and it's picked up by cells that have receptors for it. If those cells are in the liver, then what it'll do is it'll cause glycogen, a polysaccharide, to be broken down into glucose. That glucose will be released. It'll raise the set point, homeostasis restored by two systems that work antagonistically to maintain a set point. Let's switch from feedback to evolutionary biology. In evolutionary biology, the meanings of positive and negative are pretty much what you'd expect. Positive mutations increase fitness. Fitness is about surviving and reproducing, whereas negative mutations decrease fitness. However, that always depends on the context. So for example, the sickle cell mutation, which is shown over here, is disastrous if you inherit two alleles. But if you inherit one allele, then you have resistance to malaria. So if your Homozygous recessive, it's a negative mutation. If you are a heterozygote in an area that suffers from the parasitic disease malaria, then it's a positive mutation. In relation to experimental design, a negative control shows what happens when the independent variable is absent, and that allows you to see the effect of the independent variable on the experimental group. Here's an example. In an experiment where you're testing the effect of tobacco smoke on rats, you would have two groups, a control group and an experimental group. Group 1 in this case is the control group, and it's going to get a negative control, no smoke whatsoever, whereas group 2 is going to be your experimental group. It's going to be exposed to your independent variable, which is cigarette smoke. That's a negative control. A positive control is a different kind of experimental design. In this case, the positive control shows what happens when a known response is expected. So if you were comparing the effect of traditional cigarettes to e-cigarettes, then you would have two groups. You'd have a control group and an experimental group, but your control group in this case would get a positive control, which is smoke from traditional cigarettes. And based on previous studies, you would anticipate what, for example, the amount of cancer might be caused in this control group, whereas group 2 is your experimental group, and that would be exposed to smoke from an e-cigarette, and you'd look at the difference. What's the takeaway? In biology, positive and negative don't equate to good and bad. It depends on the context, and you need to think carefully about that. That might relate to feedback and homeostasis, evolution, or experimental design. Pitfall number 6. In an organism, all cells have the same genes. Different tissues are different because they express different genes. Why are muscle cells and nerve cells different? It's not because these cells have different genes. The DNA within each of these cells is the same. Almost all cells in the body of a multicellular organism are genomically equivalent, and that includes you. The cells in your skin are equivalent to the cells in your heart, which are equivalent to the cells in your liver in terms of their chromosomes and their DNA. We know that to be true because of the phenomenon of cloning. You can take a cell from any part of a cloning target's body. You can then fuse that extracted cell with an egg from a donor. That egg will have its nucleus removed so it has no genetic material, and then you can stimulate that fused cell to become a zygote. It's got a full array of chromosomes. That zygote will develop into an embryo. That embryo gets implanted into a surrogate mom, and when that surrogate mom gives birth, this cloned organism is genetically identical to the organism that had its tissue removed in order to start the entire process. The only exceptions to that are gametes. Because of meiosis, gametes are genetically unique and distinct from the rest of the cells of the body. Cells differentiate because they express different genes. Even though those cells might wind up to be phenotypically extremely different, as with a nerve cell or a muscle cell, they are all descended from the same zygote, and they all have all of the DNA that zygote received from the gametes that formed it. As that zygote develops through mitosis, certain genes are going to be activated in the cell that will become a neuron. Other genes will be silenced. Other genes will be activated that are in the cell that will become muscle tissue, and other genes will be silenced. And as a result, you'll have cells that are phenotypically different, but they're different only because they're expressing different genes. They have the same DNA. Gene expression is about epigenetics. And among other processes, DNA is shut down through the process of methylation. That means the attachment of a methyl group to a DNA base, adenine, thymine, cytosine, guanine. And DNA is conversely made available for transcription by acetylation of histone proteins. Those histones are the proteins that DNA gets wrapped around when it's organized into chromatin, which is what gets organized into chromosomes. So again, Epigenetics is how different tissues are different, not genetics. What's the takeaway? All cells have the same genes, except for gametes. Tissues differ because of differences in gene expression, and that comes down to which genes are turned off and on, and the difference between any two different tissue types with different phenotypes is not about their genes. They have the same DNA. It's an epigenetic difference. It's a difference in DNA packaging. Pitfall number 7. Dominant alleles are not necessarily common and they don't increase over time. This is a pitfall that I've seen some of my best students fall into because the word dominant somehow is thought to mean stronger, and you think that stronger things will increase over time, but that's not the case. Let's start with some basic genetics review. What's the difference between dominant and recessive alleles? Dominant alleles are always observed in the phenotype of the offspring. We represent them by a capital letter, capital A in the Mendelian system. Recessive alleles only show up in an organism that's a homozygote, and they recede into the background when paired with a dominant allele. So here we have a cross between a homozygous dominant and a homozygous recessive over here. All of these offspring are heterozygotes. None of them show the recessive phenotype. All of them show the dominant phenotype. We just discussed what you solve for with a Punnett square, what you follow with a pedigree like the one shown here, is all related to genetics. Genetics is about the transmission of traits between generations. Population genetics is about the frequency of alleles within a population's gene pool. And just to review that term, a gene pool is all of the alleles of all of the genes in a population. The pitfall that we're trying to avoid has to do with this misconception, which in my experience is the biggest misconception that AP Bio students have related to population genetics. That's the wrong idea. That's why it's crossed out, that the dominant allele has to be more common than the recessive allele. That idea is wrong. The frequency of an allele has nothing to do with whether it's dominant or recessive. Allele frequency, rather, is based on the advantage or harm that an allele confers to its associated phenotype or random historical factors. To think about this in terms of population genetics, it's about natural selection or it's about genetic drift and history. An example of this is that the allele for achondroplasia, which is a phenotype that the actor Kyle Pasek has, and Kyle, forgive me if I'm mispronouncing your name, is exceedingly rare. The frequency of this allele in the population is very, very low, but it's a dominant allele. But it's not spreading and it's not common. As we've seen, dominant alleles can be exceedingly rare, and recessive alleles in a gene pool can be common. This is the 2015 Norwegian women's soccer team. And you can see that the blonde hair phenotype is loosely recessive. Of course, blonde hair, blue eyes, they're actually controlled by multiple genes, but we'll just think about them using a very simple model for now. But it's estimated that 50 to 80% of Norwegians have blonde hair, a recessive allele at very high frequency. Why is that? Again, recessive alleles don't have to be rare. Dominant alleles don't have to be common. Their frequency within a gene pool is based on things like evolution, and skin tone is an evolutionary adaptation. Dark pigmentation, which you find close to the equator, protects against cancer-causing ultraviolet radiation, and it protects folic acid, and that's a vitamin needed for healthy embryonic and fetal development. Light pigmentation, which is what we find in the northernmost areas of the world, with a couple of exceptions, allows skin to synthesize more vitamin D, and that's needed for healthy skeletal development. So again, recessive alleles can be common based on evolutionary factors, and dominant alleles can be common, but not because they're dominant, because of historical factors like evolution. So what's the takeaway? Just avoid this mistaken idea that dominant alleles have to be common or will grow over time. The fact is that whether an allele is dominant or recessive is about genetics, it's about inheritance. Whether an allele is frequent or rare is about the evolution of gene pools, and the frequency of an allele has nothing to do with whether it's dominant or recessive. Are you asking yourself, how am I going to get a 4 or a 5 on the AP Bio exam? It's a good question because it's a hard test, but we have a plan for your success. Go to learn-biology.com and complete our interactive tutorials and interactive AP Bio exam reviews. We guarantee you a 4 or a 5 on the AP Bio exam. See you on learn-biology.com. Pitfall number 8. Evolutionary relatedness is only about recency of common ancestry. This pitfall is about interpretation of phylogenetic trees. Since this pitfall is about phylogenetic trees, let's do a little phylogenetic trees 101. Phylogenetic trees show evolutionary relationships. A key concept related to phylogeny is the idea of a clade. And a clade is a group of organisms that consists of a common ancestor and all of that ancestor's descendants. Clade is defined by a shared derived character, a trait that identifies the clade. And it evolved in the ancestor of the clade, and it sets it apart from all other clades. So rats and gorillas are a clade. They are part of the mammal clade, and the shared derived character of the mammal clade is fur and mammary glands. Gorillas, rats, robins, alligators, and lizards also constitute a clade, and that clade has a bunch of different ancestral features, but the one that's being shown here are claws or nails. That's the feature that separates these members of this clade from organisms that are outside this clade. If you included the clade that goes from frogs down to gorillas, their ancestral feature is lungs and 4 limbs. Those are also known as tetrapods, which are distinguished from the fish, which are outside of that clade. Evolutionary biology is reflected in phylogenetic trees. Evolutionary relatedness is based solely on the recency of common ancestry. So rats and gorillas have a common ancestor over here. Rats and gorillas are more closely related to one another than rats and robins, because rats and robins have a common ancestor all the way back here at D. Rats and frogs have a common ancestor even further back in time at C, and rats and salmon have one even further back in time at B. The biggest mistake to avoid in phylogenetic analysis, the pitfall that we're trying to avoid, is this misconception. That's why it's crossed out. Vertical closeness in a horizontal tree like this one does not indicate evolutionary closeness. The frog is equally related to the lizard and to the gorilla, even though the frog on this tree is right next to the lizard, and that's because the nodes can rotate. What do I mean by that? You have to think of phylogenetic trees as like the kind of sculpture that's called a mobile. They can basically rotate around any of the nodes and change positions. In other words, these two phylogenetic trees are exactly equivalent. In this one, the frog is right next to the lizard, but that doesn't really mean anything in terms of closeness because it's based on common ancestry. The frog and the lizard, their common ancestor is all the way back here at C. In this tree, here's the frog and here's the lizard very far away, but they're still equally related. Why? Because their relatedness is based on their recency of common ancestry, and that hasn't changed. Their common ancestor is still at C. And you can make a similar analysis for any other pair of organisms in this tree. And just to drive this home, let's do a little analysis of common ancestry based on another phylogenetic tree. The American black bear and the Asian black bear, they share a common ancestor over here at C, which means the Asian black bear is equally related to the brown bear as it is to the American black bear. There's no difference. The spectacled bear and the sun bear, not particularly closely related because their common ancestor is all the way back here at B, and this is a phylogenetic tree that's rooted in time. So you can see that the most recent common ancestor between the spectacled bear and the sun bear is somewhat before 5.3 million years ago, whereas the polar bear and the brown bear split apart only 100,000 or so years ago. Same thinking applies to vertically oriented trees. So on this tree, not closely related animals include the bat and the rabbit. Why? Because their common ancestor is all the way down here. The squirrel and the galago, their common ancestor is all the way down here. The lemur and the owl monkey, they have a common ancestor over here. But closely related animals would include a dog and a cat. Here's their recent common ancestor, a mouse and a rat, because they have a recent common ancestor right over here, and a human and a gorilla. Again, you're looking for recency of common ancestry to assess relatedness. What's the takeaway? If you're analyzing a phylogenetic tree, use one criteria only, that's recency of common ancestry, to assess evolutionary relatedness. You're going to ignore the closeness on this dimension. All you're going to think about is how recently there was a common ancestor. Pitfall number 9. Energy dissipates and can't be recycled. Explain this pitfall, I need to introduce a concept that's slightly outside of the bounds of AP Bio. It's the idea of free energy. Free energy is the amount of energy in a system that's available to drive work. It's represented by this symbol over here, negative delta G, and free energy, negative delta G, drives things like dehydration, synthesis, monomers into polymers, cell division, growth, movement, repair, reproduction, basically all the things that make life possible. Free energy is required for endergonic reactions, and the way that life handles the issue of free energy is by coupling exergonic reactions, which release energy, to endergonic reactions, which require energy. Almost all the free energy in living systems originates with the sun. Photosynthesis converts low energy carbon dioxide into high energy carbohydrates. And here's the formula here. CO2 plus water driven by light energy creates glucose and oxygen. We've seen in this video and throughout your AP Bio course, sunlight provides the energy that producers use to carry out photosynthesis, and that forms the basis of all food webs in all ecosystems, with one tiny exception, those undersea hydrothermal vents. But free energy dissipates as it flows through ecosystems, and it can't be recycled. 90% of the useful energy is lost as it passes from one feeding level, one trophic level to the next. That's the 10% rule in ecology. Much of that energy dissipates away as heat, and the result is that carnivores are rare, herbivores are common, and plants are abundant. That is all about energy dissipating as it moves through the trophic levels of an ecosystem. But while energy can't get recycled, matter can. And we see that in biogeochemical cycles like the carbon cycle, which shows how carbon will move from one reservoir to another reservoir through various fluxes like photosynthesis and respiration. If you want the details about the carbon cycle, we have a tutorial about that on learn-biology.com. My food chain song captures this idea. And if you know this about food chains, you can really take the cake. When energy goes through, it doesn't last, it dissipates. But nutrients and food chains meet a very different fate. They recycle and they circulate forever. Ain't that great? What's the takeaway? Energy flows through ecosystems and dissipates. It cannot be recycled. Matter does get recycled. Keep that idea in mind on the AP Bio exam and on your teacher's next test. Here are your next moves for AP Bio success. Please subscribe to learn-biology.com and please watch this next video.